Hello, I'm Suzanne Sandmeyer. I'm a professor in the Department of Biological Chemistry in the School of Medicine at UCI. And what we'll be talking about today is technological developments that are changing the way that you will be doing medicine when you graduate. In 2001, a draft of the human genome was completed, and using that draft, we've begun to understand that individual variation in human genomes can affect how individuals respond pharmacologically and how they react to the environment. In fact, what we see is that individuals differ widely by single nucleotide polymorphisms and actually by blocks of sequence in their DNA, and these are normal individuals. So today what we'll be doing is examining one of the technologies that is going to introduce personalized medicine to you when you're out in the clinic beginning to practice, and we'll see exactly how that's going to come to pass. So with that introduction, let's go ahead and unpack that technology in the UCI Genomics High-Throughput Facility. So we're here now in the Genomic High-Throughput Facility, and standing with me is Dr. Melanie Oakes, who's the manager of the facility. And we're going to basically walk our way through the steps that we would take in preparing and actually executing high-throughput DNA sequencing on a genomic DNA sample. So that process would begin with a client of the facility bringing in a sample which would have been already purified and extracted. And we would begin then by quantitating the DNA and confirming that the sample is as pure as what we need in order for it to be sequenced in our facility. So subsequent to that, we would come to this station where we would fragment the DNA in order to make a library. So a library is basically a collection of fragments that are representative of the entire sample. And it's important that the library be of a certain size so that it can be processed consistently by the steps that we have arranged to take for the sequencing to occur. So that fragmentation in our case begins with a very nice instrument called the Covaris, which operates at 500 to 1,000 hertz in order to fragment the DNA. And Melanie is just going to illustrate removal of the probe, and this probe is what is actually generating the frequency that fragments the DNA. And from here, we'll go on into the facility where we'll confirm that the fragmentation of the DNA was appropriate and the fragments are of the correct size. So we've taken our DNA that we fragmented on the Covaris, and we've come over here to the Agilent 2100 bioanalyzer. And Dr. Oakes is going to illustrate how we load those fragments on the bioanalyzer. And the point of this step is to determine the size of the fragments that we generated on the Covaris. And that's very important because we'll be going on to describe the processing of those fragments. And it's important that they are uniform and that they're in a size range of 100 to 200 base pairs, depending on the exact type of sequencing that we'll be doing. So we use the Agilent because it requires very small sample amounts, and the samples at this point are quite precious. And it's basically a capillary electrophoresis system which is contained right within that chip. And we can electrophorese about 12 samples on that simultaneously. So this displays the electropherogram that we get off the Agilent chip. Now remember that DNA is negatively charged, so it will migrate toward the positive pole during electrophoresis. And fortunately for us, DNA is uniformly charged, so it will migrate according to its size, with smaller fragments migrating faster. And so we can see that the DNA fragments migrate in a certain pattern, and we can also see here that the fragmentation was quite effective, so we have basically a peak of fairly uniformly sized molecules, which is exactly what we want to proceed to the next step. So we'll have a PowerPoint now of the processing steps so that we're all on the same page with what's going on molecularly, but essentially we'll be adding sequences of a known identity to the ends of the fragments so that we'll be able to use primers and proceed with a synthesis-based sequencing step. And so we'll go now into the sequencing facility to examine how we do the sequencing. We've moved deeper into the sequencing facility now, and up to this point, the steps that we followed are ones that are generalizable to many kinds of next generation sequencing. But at this point, we're going to be describing sequencing preparation and actual sequencing using the Illumina HiSeq 2000. It's one way of sequencing by synthesis. So we begin that process. Melanie is holding a flow cell. The HiSeq 2000 actually contains 2 flow cells which can perform sequencing independently. Each flow cell contains 8 lanes. So we can run a total of 16 independent lanes of sequence. And each lane generates about 10 billion bases of DNA sequence. So our processing on the flow cell begins with this instrument, the CBOT. And the step that we do here is intrinsic to the Illumina process. So we have actually a lawn of oligonucleotides on the surface of the flow cell. And those oligonucleotides have sequences which have been chosen to be complementary to those ends that we just told you about we attached to the DNA fragments, so that we know the sequence of the ends and we know the sequence of the oligos. So these DNA fragments actually will stick down on the lawn that's established on the flow cell. And those DNA fragments are loaded at a very specific concentration so that they're spread over the flow cell as evenly as possible, and they are not too dense. And that's important, as I'm sure you will see in a minute. So we undergo rounds of in situ PCR so that each individual molecule, which is loaded on the surface of the lane in the flow cell, is actually amplified right in the vicinity of that particular molecule, so that you wind up with a small cluster of DNAs which all have the same sequence. And those are the clusters where we are going to monitor the sequence by synthesis. So Melanie will now show where the actual flow cell fits into the CBOT. And the process that we do on the CBOT, as I indicated, is this amplification in situ. And so these interesting tubes that Melanie is showing going into the flow cell represent the tubing that we use to pump reagents into the flow cell and out of the flow cell to support the DNA synthesis or in situ PCR polymerase chain reactions that we're using to do this amplification or cluster generation on the flow cell. So once we've generated clusters on the flow cell, we'll proceed over to the Illumina HiSeq instrument itself, where the sequencing is performed. We're standing over next to the HiSeq 2000 now, and the HiSeq is actually in operation, so we're not going to be able to open it up and actually show you the inside workings. But we take that flow cell that we just performed cluster synthesis on, and that is now loaded into the HiSeq. So the flow cell is undergoing cycles of DNA synthesis, and we're being unable to do that because reagents are being pumped from reservoirs down here behind this panel by these pumps that are displayed up here. And so there are cycles of reagents going into the flow cell, being pumped out, washes, and replacement with new reagents. So now we're going to get into a little bit of detail about exactly how the sequencing works. So a key aspect of the Illumina sequencing is that they were able to develop fluorescently tagged nucleotides which could not be further extended. So with each cycle of the sequencer, where we would gain the identity of one base for each of those many clusters that we formed on the cluster station, nucleotides will be flooded in. As you know, there are 4 nucleotides, G, A, T, and C. We will abbreviate them. And each of these is different colored with a fluorescent tag. And what's important about these nucleotides as well is that they cannot be extended. So every cycle of sequencing, at each position on the flow cell, there will be a particular colored nucleotide incorporated. Now, at the end of that cycle, a CCD camera, not unlike the camera that you may have at home, scans over the flow cell and records the color at each position. And because those positions do not change as the DNAs and the clusters are extended, we can use computers at the end to compile the final sequence of DNA molecules at each position. So Melanie will now take over and will describe some of the information that actually is generated in real time for us to monitor the progress of the sequence while the sequencer is running. And that's important because if we do what we would call a paired end 100 cycle sequencing, which means that we sequence first 100 cycles from one end of the DNA and second 100 cycles from the other end of the DNA, that process actually takes about 10 days. And so we need to know that that process is continuing correctly during that time. So Melanie will take over now and describe the output from the information screen.